Let's talk a little bit about the challenges of drug design and drug discovery. Humanity, that's us, invests about $300 billion every year in drug development. This is second only to our spending on wars, right? That's a lot of money. So one can ask, what do we get in exchange to this huge investment? And obviously we are allowed to ask this question because this is money that is coming from us. You know, it's big public companies that are funded by our pension funds. So this is money that is actually paid by us. So it's really legitimate to ask, what do we gain from this huge spending? And the answer is not much, actually. We get about 40 new drugs every year. Now, this could have been a lot if these new drugs would have been breakthrough drugs. But if you log on now to clinicaltrials.gov or to the site of the FDA, and you want to see, the FDA has a table of the drugs that have been approved recently. So you can sift through the pile of approvals, and you will see what kind of new drugs we're getting in exchange to our money. So this could be drugs that extend the survival, the average survival of cancer patients by 2 months. You know, it's not nothing, but it's not breakthrough, right? Or it could be a drug that has been modified such that instead of being given in an IV infusion, you can give it in an injection. That could be considered a new drug. It's much easier, you know, instead of sitting on a chair for an hour, you can get it in like a minute. But it's not a revolution. So if you look at an analysis of new drugs, and this is an analysis of actually almost 1,000 new drugs that were studied in this analysis, and the question was, is the drug completely changing the way we treat a disease? In this particular case, the question was, do we give the patients more than 1 year with respect to the previous treatment? And the answer is that in 90-something% of the drugs, the new drug was not significantly better in terms of what it gave the patients with respect to the previous drug. Only 2 drugs were found to be breakthrough drugs based on the definition I just gave. So we're spending tons of money, most of the effort fail, and the efforts that do culminate in an approval, namely the so-called successful attempt, give us drugs that are usually not breakthrough drugs. So what is it that we're doing wrong? You know, around the table for developing new drugs, there are 3 players. Science sits there. Money sits there, investors. And the third player is, of course, the regulator, the government, right? In order to develop new drug, you need to have understanding of the science, you need to have funding, and you need to have the regulator that tells you you're doing the right thing. Now, if you ask any of these 3 players, why are we doing so poorly? They will all have the same answer. It's because of them, right? And the truth is that they're all right. The science does not give us good enough drugs. The regulator is very, very careful and puts a lot of roadblock to development. And the investors are very hesitant because normally they lose money. So they're very reluctant to put money on revolutionary ideas. I gave this intro to the world in which we're acting now just to give you a glimpse of how difficult it is to make changes in this world. But what I want to try and convince you is that AI can remove many, many of the blocks and hurdles that we have in our way to design new drugs. And I want to give you a bunch of examples from things we have been doing over the last years to cope with these challenges. Remember that, as we said, there are 3 players. Let's focus on the science. In order to develop new drugs, we need 3 things. Number 1, we need to understand biology in a way that will help us understand what we want to attack. Second, we need ammunition. We need to know with what molecule we want to attack the target. And third, we need to know which patients we want to treat. In all 3 fronts, we face great challenges. I want to start with number 3. Which patients should we treat with which drugs? And I want to show you some quite simple example for a study we did many years ago that suggests a new way of addressing this problem. One resource that we have here in Israel that very few countries have is public health care. Every Israeli citizen, from cradle to death, is covered by usually one insurer, Holon. So there is an organization with an electronic file of all the doctor visits, all the diagnoses you got, and all the prescriptions you got. And we thought that maybe if we can get access to these files, we can find some new insights into which drugs can cure which disease. So this is what we want, what we did. We went to Kupat Holon and we said, can we get the medical records of all the cancer patients you have on file? Of course, we can't get the names, we can't get personal details, but we can get everything else that is anonymous, that will not allow us to identify the patient. So if you work with the right team inside Kupat Holon and you write the right check, you will get the data. And actually, the government here in Israel wants to make it easier for researchers to get these data. And now what we wanted to do is to look for a new drug for cancer. Everybody wants to do that. But here is what we tried to do. We looked at the files and we asked, can we find a drug that many of these patients take and helps their survival? That's easy, of course. Everyone looks at cancer drugs and wants to check whether patients are doing better. But that's not what we tried to do. We looked for drugs that are not cancer drugs. We asked, maybe they take something for their skin, and patients that take the drug for skin do better than patients that don't take the drug for skin. So basically what we did is something that is called data mining. We went to the data. We didn't have any hypothesis. We did not know what we're looking for. What we searched is one drug that some cancer patients take, not because of their cancer, and some cancer patients don't take. And we want to see whether the population that takes that drug is doing better than population that does not. And what you see here is a plot that is very common, very popular in cancer research. And this plot is called the Kaplan-Meier plot. And basically what you see here is the survival of patients over time. So the first day of the study, we have 100% of the patients. And unfortunately, over time, they die. Okay? And you see here 2 lines, right? What we did is we looked for patients that take a specific anti-diabetic drugs. So some patients here have diabetes, and they take a drug for the diabetes, a very common inexpensive drug that costs something like 20 shekel. It's a very common drug, out of patent, everybody can get it. What we saw is that the survival of patients that take this drug is better than the survival of patients that don't take the drug. Now, to be very careful, what we did is to look for what people call epidemiological twins. So for every patient that takes metformin, the drug for diabetes, we looked for another patient of the same age, with the same type of cancer, and with diabetes that does not take the drug. And what this shows you is that even when we were very careful with our controls, we were able to generate an interesting hypothesis, because you see that the patients that took the drug on average did better than the patients that did not take it. So this is something, if you have the right algorithm to sift through the data, you may be able to find new ideas. And now, this specific example is obviously not a clinical trial, because we didn't give the drug to patients and waited to see what happened. We just looked at the data. But I can tell you that, first of all, we did it about 10 years ago. Since then, many researchers did similar research and found similar results. And I can tell you that based on these studies, some oncologists already recommend to their patients to take this drug, even if they don't have diabetes. I don't know if this is a generally accepted view, but here is one idea of how we can use data mining, and machine learning to come up with new ideas on how to develop drugs. So this example actually suggests that we can maybe explore existing drugs and find new applications to the existing drugs. But can we actually develop new drugs using AI, or even design new drugs? So I want to tell you about a study that was led by a graduate student in my lab, Ariel Feiglin. He later graduated and went to Harvard for a postdoc, and then came back here and started a very exciting startup company. But back when he was in his PhD, he asked the question that I just asked. Can we use machine learning to design better drugs? So in order to understand what he did, we need to understand one basic thing about drugs. Drugs, almost all drugs, are molecules that bind proteins. There are very few exceptions to that. Proteins are the nanomachines that execute life. Everything that happens in life is mediated by proteins. You know, you've seen these movies of a sperm cell swimming up to the egg. It has the flagellum in the back, a propeller הזה. What rotates it? It is rotated by a fascinating and an amazing nano engine that rotates at 500 RPM. And this nano engine is a protein. When we eat food, we say that we digest the food. What breaks up the molecules of the food and extract energy for them? Proteins. What gives the color of the hair or of the eye? Proteins. Everything that happens in life is mediated through proteins. Drugs are molecules that bind to these proteins and help them do something or disrupt their function, usually disrupt. So when we want to design a new drug, we want to design a molecule that will bind a certain protein and will stop it from doing what it does. What do people usually do when they search for such drug? They screen billions and billions of molecules, one after the other, until they find one that works. This can take years, and most often it fails, because either you don't find the right molecule, or the molecule you find is not stable enough, is too expensive, and so forth. So what Ariel wanted to ask is, can we computationally, using machine learning, come up with drugs that will bind the target protein Now, as you can see here, we can have multiple molecules, and basically the question is, which molecule will bind this target in a way that will give us the medical outcome that we want? You can computationally take a molecule, take a protein, and try to fit them spatially to each other, like so. You see 3 molecules. We can fit the purple one here, we can fit the blue one here, we can fit the red one here. Perfect. But these interactions do not happen in reality. Actually, out of these 3, just this one happens in reality. If we put in solution these 3 molecules with this protein, the purple will not bind here, the red will not bind here, but the blue will. How can we know what's going to happen in reality? So what Ariel did was basically to take all the examples in which we know experimentally which molecule binds which drug and train a machine learning model on the positive examples, pairs that do interact, and on the negative examples, pairs that do not interact. I will not drag you through the mathematical details, but computationally this looked very good. Ariel was able to generate the data set computationally. So he was able to predict correctly on the existing data set which molecule will bind the drug and which will not. So on paper, it looked wonderful. But the question is, can we actually make a new drug this way? We were thinking of ways to prove what we do until Mira Bardasad, an immunologist from Bar-Ilan University, contacted us and said, look, I heard that you have a computational method for designing new drugs. I can tell you that in my immunology research, said Mira, I found a protein called WASP, and I think this protein is actually the protein that drives metastasis, drives גרורות של סרטן. I think that if I find a drug that stops this protein, cancer cells will not be able to migrate to new tissues and create metastasis. But I can't find a molecule that does that. I screened, and I couldn't find one. Can you computationally find a molecule for me? I said to Ariel, that's the opportunity. Sit down with your algorithm, find a molecule for Mira. So what Ariel did was very elegant. He took a data set of existing molecules, 1 million molecules. What was special about the data set that he took is that each of these molecules had a link that allowed you to click on it and to buy the molecule. So these were molecules that someone somewhere around the world knows how to synthesize. He used his algorithm to try and predict which of these 1 million molecules will bind Mira's protein. And he actually found 14 molecules. He sent the links to Mira, and Mira bought the molecules. 11 of the 14 molecules actually bound the protein that Mira was interested in. So what Mira did was to take cancer cells, as you can see here, and put them in 2 plates. In this plate, she put one of the molecules that Ariel found computationally, here, and in this, she didn't put anything. And now you can see how the cells, without treatment, migrate, they move. And the cells that got Ariel's molecule, they don't move. So Mira was able to show that the molecule that Ariel found using machine learning actually stops the movement of cancer cells. That's very exciting. Will it actually help cancer? So Mira went on and tried to treat mice with these molecules. So these are results of treatment. You see here 6 treatments with a molecule that was found by AI. This is the volume of the tumor for mice that were not treated, that you see that after 2 weeks, the tumor grew really fast. But when the mice were treated with the computationally identified drug, the tumor was very significantly inhibited. So here is a journey from looking at molecules, training a machine learning model, to the point where you can actually see in animals that the drug may do something beneficial. This molecule is now in clinical development, and hopefully it will get into patients soon. One example of what you can do with AI to design drugs. That's example number 2. Now, what Ariel did was to work with small molecules. You remember we looked at very small molecules when we compared them to the protein. You know, the protein, this is the protein, it's quite large, and the molecules are quite small. But some of the best drugs that we have today are not small molecules, like this one, but are very large molecule, like this one. This is an antibody. And actually, from among the 10 best-selling drugs, the drugs that make most money in the market, about half of them are antibodies. Why is that? Because antibodies are human molecules. We all have antibodies. And if we can find an antibody that somehow inhibits the function of a protein, that could be a wonderful drug. So some of the drugs you hear about in the news, Herceptin, Keytruda, some of the drugs that made the biggest splash in the context of COVID, they were all antibodies. So we ask ourselves, can we actually use AI to design better drugs with new capabilities? Drugs that will do things that traditional drugs can't do. So I'm going now to tell you about work that started in a company that we started about 10 years ago. It's a company called Biologic Design. In Biologic Design, we tried to use machine learning and AI to design smarter drugs. Last year, we started treating patients in Australia and in the US for the first time ever with an antibody that was designed computationally. So this company from Rehovot, an Israeli company, designed the first antibody that was computationally designed to get into patients. I will not tell you the story of that antibody. Maybe some other time. We're seeing really exciting results from these cancer patients. But I will tell you about some new programs that we have at Biologic Design. So, as I said, usually drugs bind to a protein and disrupt its function. Can we do things that are more creative with antibodies? Here is the idea we wanted to try. We said, can we come up with a human antibody that will have two functions? Let's say an antibody that can bind both chemotherapy and it can bind tumors. Why is this interesting? So imagine that we have a patient and the patient has a tumor, you see? So the patient gets chemotherapy, and the chemotherapy is usually evenly distributed all over the body of the patient. Where do we want the chemotherapy? We want it here. Where does it go? Everywhere. That's very, very bad. It's bad not just because the patient suffers. It's bad because that due to the effect of the chemotherapy everywhere else, we are limited by the amount of chemotherapy that we can give. If we could have given chemotherapy that will go only here, we would be able to give much more chemotherapy that will kill more tumor cells and will have greater chances to cure the patient with less side effects. So we said, can we give this patient an antibody that binds chemotherapy in the periphery and releases it in the tumor microenvironment? Imagine that the patient gets an antibody, then the patient gets chemotherapy. The antibody grabs the chemotherapy, doesn't let the chemotherapy do any damage in the periphery, and concentrates it to the tumor. That's a very nice fantasy. But what do we need to be able to do in order to allow for that? Actually, if you learn some chemistry, you understand that this may be possible if we allow for simple competition. What does competition mean? Can we design an antibody that can bind both the chemotherapy, let's call the chemotherapy molecule A, and something on the tumor, let's call it protein B? If we can design an antibody that can do both, it means that in any given point in time, each arm of the antibody can bind either A or B. Okay? Now, if B exists only in the tumor microenvironment, what would happen when the antibody is in the periphery? It doesn't see B. So what will it bind? A. So it will grab the chemotherapy, sequester it, and won't let it do any harm to the patient. Now, what will happen when this antibody comes with two chemotherapy molecules and gets, by chance, to the tumor microenvironment? All of a sudden, it sees who? B. It likes B. Now we start simple competition between A and B over this site. Some of the antibodies will eventually release A and bind B. So if we are able to design an antibody that can bind two things through one arm, we may be able to come up with this mechanism of molecular exchangers. An antibody that grabs chemotherapy from the periphery and releases it in the tumor microenvironment. So I want to show you how we are approaching it and what kind of machine learning examples or machine learning models we are using to address this problem. An important fact to remember. You may have seen today some examples of machine learning, so you already know that the most important thing for machine learning is data. If you want to distinguish between cats and dogs, you need millions of pictures of cats and millions of pictures of dogs, and you train the algorithm, and eventually the algorithm will be able to distinguish between cats and dogs. We want an antibody that can bind A and B. We need millions of examples of what it takes to bind A and millions of examples of what it takes to bind B. They don't exist. So what we have to do is actually to generate the data for our own algorithms. And this is why biologic design has a strong computational team, but an even larger team of experimental biologists, because they need to generate the data. So we went and performed experiments on what it takes to bind A and what it takes to bind B. I want to show you some predictions. So what you see here is measurements of molecules that we use to train in order to bind target A. So this is the score we got from the prediction for how tightly we bind A, and this is the observed binding of A to the antibody. You can see that there is a very strong correlation. This is on the train set. On the test set, you can see that the correlation is a bit weaker, but still very strong. If we take only these antibodies here, we get antibodies that get very strong predictions, and they actually bind A. We can do the same thing for B. You can see here predictions and observation on the train set for binding target B. This is on the test set. You can see that we can very accurately identify antibodies that bind B. So if we run these algorithms on billions and billions and billions of sequences, we will eventually be able to find an antibody that binds both A and B. And that's what we want, right? No, that's not enough. We need this antibody to also be a drug-like antibody. What does it mean? It's not enough to solve the scientific problem. The drug has to be a product. What does it mean? It means that we need to make it in a factory somewhere, in this example, in China. Then we need to put it on a boat, ship it to a hospital in Australia. We need that over time, on the boat, the antibody will stay intact, will not decompose. Then you put it on a truck. Then you get it to the clinic. You freeze it, you thaw it, you freeze it again. It has to be very stable. If you get an excellent antibody that in the lab does what we described, but if you make it as a product, it decomposes after 1 hour or even after 1 week, you don't have a drug. So we had to train another model that predicts how stable the antibody is. And you can see here some measurement of the stability. And you can see that, again, there is a good correlation between the prediction and the measurements, which means that now we have 3 machine learning models. One that tells us whether the protein binds target A, say the chemotherapy. One that tells us whether the protein binds target B, say something in the tumor microenvironment. And a third model that tells us whether the antibody we found can be a drug. So now you realize that if you have this combination of understanding what you want to achieve, being able to generate the right data, machine learning can actually help you design drugs that do things that existing drugs today will not be able to do. If someone uses traditional methods in search of an antibody that will bind chemotherapy and a tumor-specific antigen and will be developable, it's the probability of finding it experimentally by chance are very, very, very slim. But we have a system that allows us to do it time and again, and we hope that this kind of antibody will be in the clinic very soon. So that's a third example. What other things can you do with machine learning when you try to design clever or smart proteins? So we saw how we can use machine learning to repurpose existing drugs. That was the first example. We saw we can use machine learning in order to actually design new small molecules to be drugs. And we saw that it works in mice. We saw that we can use machine learning to design antibodies, to design more sophisticated drugs than we actually use today. I want to talk about another effort, and that's the effort of designing food proteins to be safe. And this is an effort that is concentrated in another company we started called UCO. So what does UCO do? UCO looks at proteins that cause food allergies, like peanut proteins that cause allergy, or like gluten that cause celiac disease. And what we ask is, can we design these proteins to be safe? Can we design peanut proteins that do not cause allergy? How is allergy develops when a patient eats peanuts? So allergy is driven by a class of antibodies called IgE antibodies. They usually sit on basophils and mast cells. Now these antibodies are natural antibodies that occur in our body, not like the antibodies we design on the computer. They occur naturally in every person. In people with allergy, these antibodies, for some reason, identify some nook or cranny, some chupchik on the peanut protein. And once they bind this determinant on the peanut protein, this initiates a cascade of events that eventually leads the cells to degranulate, release some compound into the blood, and you see an allergy. It has effect on the respiratory system, on the skin. Patients can die from it. We asked, can we take this protein and design it such that it keeps all its characters? It's a normal peanut protein, but the IgE antibodies cannot bind it.
When you say design, you mean synthesize or in nature?
So we want to take the gene that encodes the protein, change it minimally, really tiny changes, and then to be able to put it back into the plant and make a new plant. Or even better, use it as a drug and give it to patients so that they're cured. You know that the only treatment for allergy that can lead to cure is exposure. Kids with allergy go to the doctor and they get tiny, tiny doses of peanuts every day. And after 2 years, some of them can tolerate peanuts. But most of them, through this process, get very, very significant allergy attacks because you give them peanuts. Can you do the same with peanut proteins that cannot cause allergy? So the answer is, after a lot of research, we found a way of doing it, at least for peanuts. And as we say, I have great news to all the mice in the audience. We were able to cure mice from their peanut allergy because we took the peanut proteins, we changed them minimally, we injected the peanut proteins to mice, and after a few weeks, we gave them peanuts, and they tolerated it very well. So obviously the next step is to try it on people, and that will happen next year. But that's one thing that you can do when you understand what and how you want to change the protein. Another example is gluten. You know that gluten causes diseases like celiac. But what does actually happen when gluten is presented to patients? So first of all, we need to remember that gluten is what makes bread fun. Some of you may have tried gluten-free bread, and it's a lot of things, but it's not as fun as bread with gluten because gluten makes the bread elastic. It makes those tiny bubbles. It makes the smell. It makes the feel of the bread. No one probably has seen a reasonable croissant without gluten because this thin dough that is elastic and is almost transparent when you hold it against the light is only through the characteristics of the protein that is called gluten. It's actually a big family of many proteins. But in celiac patients, when they eat gluten, the immune system attacks the gluten and leads to inflammation of the intestines, and they cannot eat gluten. So our question was, can we take the gluten and change it minimally in a way that it is no longer identified by the immune system, but it still makes a fun bread? So we collected a lot of samples from patients. We engineered the gluten, and we saw on the samples from patients that after we engineered the gluten, it did not irritate the immune system of the patients. So we were able to change the gluten in a way that it is probably going to be safe for patients. But is it still gluten? Maybe we changed it too much so that it doesn't make the bread fun. So we did some baking experiments. We took the gluten that we engineered and we added it to gluten-free flour. This is what happens when you bake with gluten-free bread. You see the bread is flatter. It's sticky in the mouth. When you take this gluten-free flour and you add to it our fixed gluten, the safe gluten that we engineered, you see that it has the color of a bread rather than this pale color. You see that it's more elastic. You see that it looks more like a bread. And I can tell you that it also tastes more like a bread. So maybe what we see here is that we were able to engineer the gluten in a way that makes it safe for people with celiac and for people with other with other gluten sensitivities, but still keeps it as a functional gluten. This is almost wonderful. Why almost? Because the gluten that we used here was made in the lab, not in wheat. We had to put it inside a cell that will make it. So this little loaf of bread is probably $100, right? So what do we need to do now? To find a way to make it on a larger scale. For example, make it in rice. Knock it into rice, grow the rice, and now the rice has gluten in it. Or even knock out the gluten from wheat, put it back into it after we fix it, and then we may have flour that is as inexpensive as normal flour. It's functional like normal flour, but it's safe for everyone. So this is actually the final example I wanted to give to you today on how we can use machine learning in ways that help us make our environment safer, the proteins that we use more effective, and the drugs that we use to cure diseases safer and more efficacious. So thank you for listening, and I'm happy to take questions if there are any. That's a great question. So what we try to do usually,
what was the question?
The question was what kind of features we're using when we train our models. Do we use just sequence? Do we use structure? Do we use some other characteristics of the proteins we describe? So it varies from one problem to another. In the example of the small molecule prediction, the data from mice I showed, we used structural data. In everything else, we used only sequence. What we're trying to do is to rely as much as we can on sequence only, because that opens the door for processing more cases and looking at more data.
A couple of things. One related to the diabetes metformin, and the second one related to the molecules for the chemotherapy. Starting with the first. You mentioned that people that took the second drug provided better
survival
prognosis of life,
yeah,
eventually. Now the question is double-sided. A, was it because they took some kind of anti-diabetic or specifically the metformin? Two, maybe it was the other way around. The very fact that they were diabetic made them more prone to earlier death, and whichever drug they took prevented death.
So these are
question one.
Yeah.
After the answer, I'd like
all
to
right. So these are great questions, and you're actually highlighting the challenges of doing this kind of retrospective analysis instead of a real clinical trial. The only thing we can do in order to try and answer the good questions you asked is we can try and make the right controls. So for example, I mentioned that we take diabetics who have cancer, both in the metformin group and in the other group, and the other patients are also treated with some drug that is not metformin. So that takes care of the concern of maybe it is different underlying or background comorbidities that the patients have. They all have the same comorbidities. We also tested it actually on patients that don't have diabetes. Metformin became a popular drug over the years, and some patients get it for other reasons. For example, some gynecologists give it to their patients who may have some a menstrual problem. So when we look at these patients who got it for diseases that are not diabetes, we still see a benefit. So obviously the only answer will come after a real clinical trial. And by the way,
Actually, one
what-
more control you might need is to check another diabetic drug on diabetic people, like insulin or something.
yeah, so the control had insulin.
In order to see if it is just whichever anti-diabetic drug or only specifically.
No, so I mentioned the first control was patients that got other diabetic drugs.
I didn't hear drugs, I heard yeah. Okay, second question related to the chemotherapy. Actually, what you're doing there, you're saying, okay, I'm giving chemotherapy which hurts the tumor but also hurts the other parts of the body. Now I'm trying to do something that corrects everything that hurts the other parts of the body and only hurts the tumor. Why not go backward and say, okay, I'm creating a drug that only hurts the tumor and is inert to everything else. I mean, not let everything else be prone to attack and collective, but never attack.
Yeah, of course, that's what we want. Easier said than done. We are actually the drug that we introduced into patients a few months ago in the clinical trial I just described is attempting to do that. It's a drug that identifies a certain immune population that is active against the tumor and expand that population very selectively. But you know, cancer is a very shrewd disease, and we need all kinds of tricks to try and overcome it, and that's one suggestion. An important thing in terms of the development path is that if you add something to chemotherapy that shows benefit, then it's easier to start from first-line treatment. That's a regulatory concern, right? If you start with something like you suggested, you always start from the last line, and then you get the patients with the most difficult to treat cancers. If you show that there is a way to make the first-line treatment more effective, you can get to patients with an earlier stage cancer faster. So there are many reasons to do different things, and we should attack on all fronts.
Thank you very much.
Thank you. Yes,
Very short. If you genetically change the cell molecule, is it possible that it somehow creates a toxic molecule?
Great question. So the question is this. People have immunological reaction to gluten. If I change it, some patient may have a celiac-like disease against my molecules. Obviously, this is possible, but how likely is it? Think about this. We eat about 5,000 different species, right? There are 5,000 different species on the planet that humanity calls food. In how many of them do we have a protein that causes a celiac-like disease? One. It happens only with gluten. So the likelihood that from the millions of other proteins that we eat, none of them causes a disease like celiac. And randomly, the one molecule that we change minimally will all of a sudden will cause that disease. It's possible, but it's highly unlikely. And the only way to know is to try. Of course, it has to go through clinical trials. Thank you, everyone.